Welcome back to part 2 of the screencast on hearing. At the end of part 1, we had talked about how the amplified vibrations from the stapes are transmitted to the perilymph of the cochlea and travel through the whole thing and back to the round window. Those sounds that you do not hear make that journey without disturbing anything inside of the cochlea. Those sounds within the frequency range that humans do hear disturb, we said, the structure called the basilar membrane as they travel through the cochlea. So now we have to better understand the cochlea, find our way to the basilar membrane, and see what's going on there to turn vibrations into neurotransmitter release. So if you take this structure here and roll it all up like a cinnamon bun, there we go, you get something that looks like this. So here's the cochlea all rolled up, and then you take a section through it, and it's multiple sections through the same, what looks like three-chambered structure. In order to find the basilar membrane, you have to zoom in a little further. So if you zoom in on just one cross section through the chambers of the cochlea, you get something that looks like this: your scala vestibuli, your scala tympani, and the cochlear duct in the middle. Lo and behold, this structure right here is our basilar membrane. So we've come back to where we left off before with sounds in your hearing range vibrate the basilar membrane at a specific location. So now we have to talk about what's on top of that basilar membrane and how those structures transduce sound or vibration into sound. So sitting on top of the basilar membrane is the organ of Corti. It is composed of hair cells, a tectorial membrane, and then the basilar membrane itself. As I said, this is where transduction of sound takes place. So here is your basilar membrane. You have a number of hair cells along with a tectorial membrane. So I am going to leave this slide up for a couple minutes and read over or talk about the text on the next slide, which should be this slide here. But we're going to leave that up. So it says these hair cells have stereocilia. So they're hairs, but we call them stereocilia. They protrude into the fluid, the endolymph, and are actually stuck into the tectorial membrane. Now you have to remember that the hair cells are receptors, which means they are modified neurons. They have the ability to alter their membrane potential in response to stimuli, which then alters neurotransmitter release. In the case of your hair cells, they're kind of a nice on-off switch. If you're not hearing anything, they're not releasing any neurotransmitter. So when a sound that you can hear vibrates your basilar membrane, it moves up and down, and when the membrane moves up and down, the hair cells get pushed up closer into the tectorial membrane, and the hairs bend because the membrane is somewhat stiff. Those hairs are hooked up to what are called mechanically gated ion channels. I believe they are sodium channels. So I will give you a second to think about what happens when you open a sodium channel on any given cell. You should say that the cell depolarizes. This is one of the things that you always want to keep in mind, is that all of your cells have that same or similar sodium potassium ATPase, which means anywhere you are in the body, if you're inside of a cell, there is low sodium, high potassium. If you're outside of a cell, it is high sodium, low potassium. So this applies to any cell, as well as the cells in your bloodstream too, right? So your blood cells are going to have very little sodium in them. There's going to be more sodium in the plasma because that's an extracellular fluid. Sorry for that digression, but understanding electrolyte balance is one of the keys to anatomy and physiology. So, Vibrations finally make it to the basilar membrane, cause the hair cells to get bent. Sodium rushes in through ion channels that get pulled open. That depolarizes this neuron, which then dumps neurotransmitter onto dendrites of our cochlear nerve, which will eventually relay that information to the auditory cortex and provide the sensation of hearing something. And so, oh, I forgot this was here. So, this slide right here summarizes everything you need to know, and there is coming up on the exam a possible open ended essay question about hearing, and you just have to describe all of the structures that vibrate and a little bit about what goes on in the cochlea. But it's all right here, so you just actually, the only thing that's not there is that the outer ear collects the sound waves and funnels them towards the tympanic membrane. But then if you look down here, all of the steps are there: malleus, incus, stapes, amplification. The oval window transmits the vibrations to the fluid in the scala vestibuli. Let's see, for B, if you can hear it, it vibrates the basilar membranes. That causes the hair cells to deflect, and that's what releases neurotransmitter. So everything is there in order for you to answer a rather lengthy essay question. But it's one big process that's mechanically connected from beginning to end. So hopefully you'll be able to tell the whole story of how hearing works, because the whole story is connected. All right, then we have a few odds and ends before we finish up. I think just two more slides. So your brain differentiates pitch, high frequency versus low frequency. The way that it does that is by tracking which part of the basilar membrane it's receiving stimulatory input from. So if you read here, the fibers that make up the basilar membrane are short and stiff near the oval window. That's these guys here. Long fibers near the apex resonate with lower frequency waves. Those are these guys here. So the way this works is that anything that has the ability to vibrate will vibrate when exposed to sound of the same frequency at which that thing would vibrate if you hit it. So imagine you have two pianos on stage. You hit high C on the first piano. The high C string on the second piano will start to vibrate when it's exposed to that same pitch. So things vibrate when they are exposed to the sound that they would make if you actually hit them or made them vibrate by some other means. So keep in mind, this isn't the length of the hairs on the hair cells, but it's protein fibers built into the structure of the basilar membrane. So some parts of the basilar membrane are tight and respond to high-pitched sounds the way a tight guitar string would make a high-pitched sound, and then other parts of the basilar membrane are looser and have longer protein fibers in them, and a longer loose guitar string is going to make a deeper sound. And so that's how your brain perceives pitch, based on which part of the basilar membrane is being activated. The auditory pathways we don't care about too much, because frankly I think they're too complicated to expect you to remember. So really what you want to know is that it starts with the cochlear nerve, which is part of the vestibulocochlear nerve. I believe that's number 8. I'm so bad with cranial nerves. And then it ends up in the auditory cortex. It makes some pit stops along the way at places that are controlling eye movement, head movement, and your shoulders, so that you have auditory reflexes. But we're not going to worry about that too much. So we're just going to think that the cochlear nerve eventually relays the information to the auditory cortex, and that's where your perception of sound takes place. And that is the end of sound. Thank you for hanging in this far.